Okay, it's just hit 3:05 CAT on my watch, and obviously I do understand that some people will be joining while I'm busy introducing for today. Good afternoon, everyone. Good morning, good evening, depending on where you are in the world. I mean, I understand that people are joining us from different parts of the world. Most of you are going to be coming from Africa, but our speaker is currently based in Europe, in Paris in particular. And I think obviously the weather differences, I mean, it's quite hot where I am in Cape Town today. So welcome to H3 Bionet's first webinar for 2022. My name is Pablo Abel-Chauke, and I'm a training and outreach coordinator based at H3 Bionet in Cape Town, South Africa, under the leadership of the amazing Professor Nicola Mulder, who's also joining us today and listening in on this webinar. And I work with a group of people. So one of my jobs is to organize monthly webinars where we invite interesting and amazing speakers from across the world to give us talks on different topics. And today I'm excited to be introducing the January webinar, which is going to be given by Dr. David Tabb, who's going to be speaking on challenges for top-down identification of proteoforms. And this talk will be for about 40 to 50 minutes. And at the end, we'll have about 10 to 15 minutes of question and answer, as well as comments and discussion as well. So please do ask questions. And just kindly, while I'm hearing people speak while I'm speaking, can you kindly mute your mics? So those are the ground rules, like a few ground rules. Kindly mute your mic if you're not speaking. If you have a question, please raise your hand. And obviously we'll give you the time to speak so that we don't disturb the speaker. So if you can make sure that you mute your mic, and that if you have a pressing question, either type it in the chat box or you raise your hand, and myself and the speaker will give you attention to make sure that you sort of get to ask your question. So those are the ground rules. Just make sure we have some order in this webinar as well, and we don't disturb a speaker. So I sent out an ad which had the bio, or rather the CV of David Tabb, and as well as the abstract as well. So I'll read a bit of the abstract and a bit of the bio as well, so that you sort of have an idea of who our speaker is. I personally first met Dr. David Tabb when he was based at Stellenbosch in South Africa. And obviously he sort of assisted us in some of the courses that we ran, particularly Introduction to Bioinformatics as well. And that's when I started knowing about him as being the uncle of proteomics. And I think obviously today we'll see why he's called the uncle of proteomics. But I'll start by reading the abstract. Bottom-up proteomics has become a standard tool for molecular biology, separating peptides via liquid chromatography en route to a tandem mass spectrometer that measures the masses of a particular peptide ions and then revealing their sequences through dissociation, followed by measurement of fragment ion masses. Measuring intact proteins through top-down methods in tandem mass spectrometry, however, has been much less widely adopted. This talk, which is going to be given today, will explore the advantages of the intact proteoform measurement for biology and examine some of the challenges that have been the barrier to its adoption. Who's David Tabb, you wonder? In the 19 years since completing his PhD, Dr. David Tabb has sought to become the trusted uncle of proteome informatics, with long-standing emphases on technology assessment and quality control for bioinformatics in tandem mass spectrometry. Dr. Tabb has been part of the USA National Cancer Institute CPTAC program, the Human Proteome Organization's Proteomics Standards Initiative, as well as the American Society of Mass Spectrometry educational programs. Having spent 5 years in Stellenbosch University in Cape Town, South Africa, which is when I met him, Dr. Tabb feels very much continuing part of Africa's story. And for the last year, however, he has been a visiting scientist with Prof. Julia team at the Institut Pasteur in Paris. Without further ado, and wasting much more time, I'd just like to give the floor to Dr. David Tabb to begin giving his presentation. Dr. David Tabb, go ahead and give your presentation, and thank you so much for honoring this invitation.
Thank you very much for the invite. H3 Africa is a really great program, and I am completely on board with the idea that African scientists should be leading and publishing the fine research that comes from the samples harvested across the continent. So I'm really grateful for the chance to help do some training to support that effort. Today I'm talking about the challenges for top-down identification of proteoforms. And although I've been a proteomics researcher for 20 years, the reality is that I'm very much a newcomer to the space of top-down proteomics. I've always expected that the proteins the protein data that I was looking at were based on digested proteins, where we've chopped them up with trypsin, and now we're looking at peptides. So looking at proteins in intact forms is quite a big challenge and creates a lot of interesting difficulties for producing the data and for interpreting them. I definitely want to thank EPIC XS, which is a program here in Europe that is funding a big network of proteomics facilities trying to improve the access to this technology. And although I've listed my affiliation here with the MS Bio unit under Julia Chameau-Rook at the Institut Pasteur, I'm still secondary faculty at Stellenbosch University, and hopefully someday I'll be able to return there. So I think that it's worthwhile to start with an assessment of bottom-up proteomics as the status quo and ask, how does top-down proteomics, what can it possibly offer that we can't already do with the shotgun proteomics that's available almost everywhere on Earth? I think it's important along the way, though, to talk about why it's much more difficult technically to produce the data volumes that we work with and how we use software to interpret them. So I'm going to give a fair bit of effort to both of those points. And finally, I wanted to close with, how can we lower the bar to entry? How can we make it easier for laboratories to get started in the space of top-down proteomics? And I'll include a little bit on the story of bottom-up proteomics. How can we increase the number of laboratories on Earth that can make use of these technologies? So I would start with what top-down proteomics uses as sort of a general workflow. And I would have to say that top-down proteomics hasn't really settled into a groove where 90% of all labs use exactly the same techniques. It's not quite like that. It's still very much a technology where individual labs develop a type of specialty within top-down, and they do things their way. And if you move to another lab also doing top-down, you may find that the instrument, the separation technology, the ionization strategy, even the denaturation process can be very different among these laboratories. So this is about as generic as you can get and still be generally relevant, I think, to how people do things routinely. We start with protein mixtures. Maybe these are wildly complex, something like a cellular lysate, or even solid tissue that you will be releasing proteins from. So that is already a wildly complex mixture. It is also possible to do top-down proteomics in biofluids. Particularly, I've been working with samples recently that were produced from milk. Milk has a very high dynamic range, as opposed to the relatively flat dynamic range that you see in something like a cell lysate. So these protein mixtures are going to have bazillions of copies of some proteins and just a very few of others. We need to denature those proteins to make them separable. And for this, we're going to use urea and other tricks along the way. But keep in mind that some laboratories will not even break the disulfide bonds within these proteins on this denaturation process. So we can expect that a lot of the structure, the secondary structure that's held in place by hydrophobicity and so on, is going to be disrupted. And yet we may not have a perfect line of amino acids to represent each protein as a denatured polypeptide. Frequently, we will see that people make use of fractionation methods, something like a gel-free box or isoelectric focusing or size exclusion chromatography. There are lots of different ways you can go about doing the separation of these polypeptides. And then we use liquid chromatography, MSMS, which relates to all of the other icons that appear on screen here. I would note that in bottom-up proteomics, we're typically working from what we call a C18 column. I'll show one of the chains that relates to that in a slide coming up. But for now, I want you to think about the fact that a complete intact polypeptide tends to have higher hydrophobicity than does a very short peptide. As a result, if you use the same separation materials for intact polypeptides,
and we did that after just 1 week, after that first 1 week.
I am hearing broken widows. Please stay on mute. Yeah, there we go. Okay, thank you. I appreciate that. So when you have a long polypeptide, it's more likely to stick to a C18 chain in the column and not come back up again. So we tend to use really short carbon chains like C4, 4 carbons, as something that we can stick the proteins to and recover them from later. When when ions are flowing out of these columns, we're charging them through electrospray ionization. They are then measured in a high resolution mass spectrometer. This is very much a constant within the top-down field. You do not see people using low resolution measurements from quadruple ion traps, for example, to do top-down proteomics. Almost everyone realizes, I think, that you must have a very highly mass accurate and high resolution instrument. Correspondingly, we see a lot of Fourier transform instruments used in this place. And we've also seen more recently the use of quadruple time of flight instruments that are somewhat cheaper. We can isolate a whole bunch of ions of a given proteoform as we've seen it. So ions of a particular protein after they've been denatured in this way. We can then use something like electron transfer dissociation to cause these proteoforms to become radicals, and they break apart into fragment ions. And then we can collect the catalog of fragment ions for a particular proteoform in a tandem mass spectrum. So this is similar to what we see in top-down proteomics, but it's not similar to what we see in bottom-up proteomics, but with some important differences along the way. No digestion step is certainly one of the key aspects. We're not using trypsin to cut these proteins up into peptides first. A term that shows up very, very frequently in proteoforms, in top-down proteomics, is the term proteoform. And I realize that may not be familiar to everybody, so I thought it'd be good to revisit this definition. You can think of a given protein coding gene as producing a protein, but that is already covering over some complexity that shows up particularly in eukaryotic systems. So you can then imagine that a gene does not give rise to a single transcript that gives rise to a single protein, but rather a gene gives rise to multiple transcripts through exon shuffling. And in a lot of cases, we find that individuals have particular sequences that differ by a nucleotide or two from the standard for that species. So we have both, we have genetic changes being introduced both through which transcript is produced from this gene and which sequence variants it contains. We then see that in the process of a polypeptide straight off a ribosome becoming a mature protein, we frequently see things like proteolysis of a signal peptide that allows that protein to get to the place in the cell where it's supposed to be. You may also have certain mutations that occur in a particular individual that lead to nonsense variants. And these nonsense variants mean that you've introduced a stop codon before where it would normally be in the wild type sequence. So we can see that sequence changes can result from both this individual's genetic material and in the splicing that results in the cell, in this particular transcript being produced by that protein coding gene. And of course, we often see that these polypeptides are marked with post-translational modifications, some of which function more or less like on and off switches. So to say that a protein is present in the cell does not mean it's active in the cell. It may be that it only becomes active when a PTM is in place. So we refer to each of these combinations of specialized sequences, truncations, and post-translational modifications as a proteoform. Dozens of proteoforms may result from an individual gene, which makes top-down proteomics quite difficult. Understanding which of these proteoforms exist in vivo is quite important. And if we think about it, it is certainly possible that in our experimental handling, we will accidentally break apart a proteoform, causing smaller versions of that protein to also be visible in the data sets. So trying to discern chemically caused proteoforms versus those that exist in the functioning cell is quite important. We want to understand the distinction between those two, if at all possible. So let's examine a couple cases of what top-down proteomics can achieve that bottom-up cannot. At the left, I'm showing a sequence for a human histone, H3.3. And I've marked a whole bunch of the amino acids in this sequence in red. This refers to the fact that when we've studied these histone sequences in particular, we see a huge number of places where a methylation or a trimethylation or an acetylation may be observed. These are three different post-translational modifications. So you can imagine that if you're using trypsin or another digestion enzyme to cut this protein sequence up into short peptides of maybe 20 amino acids or so, you could see that you would lose connectivity. That if there's a post-translational modification at the N-terminus of the protein, and there's another post-translational modification at the C-terminus of the protein, your ability to discern whether they co-occur or occur separately is going to be lost if you digest this protein, because it may be that a doubly methylated version of the proteoform is the only one that takes on a particular biological activity. But because you're seeing the peptides separately, you don't really have that information that they always occur together. So which combination of PTMs enables a particular biological activity really benefits from our looking at the protein entire, not in the short peptide segments. So that is one of the big things that's caused top-down proteomics to be considered so very useful in the space of epigenetics, because understanding histone modifications helps us to understand which genes are active or inactive in a cell. Next, let's consider the problem of transcripts. I already mentioned that exon shuffling can mean different transcripts are derived from the very same gene. In this case, we are looking at a paper from 2017 in which the PD-LIM gene has a whole bunch of different transcripts that result in Enigma 1 or Enigma 2 or Enigma 3. If you were to look at the proteins that result from these different transcripts through bottom-up proteomics, you would be digesting them down to relatively short, say 20 amino acid segments. And those 20 amino acid segments are very unlikely to stretch all the way from the N-terminus to the C-terminus of this protein. As a result, if you deal with bottom-up proteomics only, your ability to discern which of these proteoforms has been observed becomes problematic. You need to be able to look at the intact proteoform to discern the entire structure. Okay. Next, let's talk about the problems of protein post-processing. A lot of proteins get truncated or clipped in their synthesis. So it could be that you just have a protein that appears full length, essentially, except that the initial methionine gets clipped off of the N-terminus. That's really, really common among sequences in the human proteome. So certainly, allowing for the possibility of that N-terminus to be clipped or not is something that we need to take into consideration. But frequently, a longer stretch of the N-terminus gets excised. A signal peptide, for example, may specify that this protein needs to go to the cell surface. And in the process of that conduct out to the cell surface, the signal peptide gets removed. I think of it a little bit like a shipping label on a package, that you don't need the shipping label once you get it to its destination. And certainly, once a protein has been folded into its mature form and probably safety pinned into this conformation by the formation of disulfide bonds, you may find that in some cases, the loops get clipped off by other enzymes that are acting on this to make it the mature form. If you don't have the intact proteoform, your ability to discern which parts of it are clipped away and not is very much diminished. So top-down proteomics is hugely valuable here. I think that a lot of people in the course of the COVID crisis have become aware that monoclonal antibody treatment is quite essential to preventing people from advancing to the need for critical care and intubation and all the awful outcomes that can result from it. So in this space, we need to be able to be sure that the antibodies we're manufacturing for use clinically are just as pure as possible. And so being able to measure the heterogeneity of therapeutic antibodies is another space where top-down proteomics has become quite helpful. I would also note that there are some folks who try to find a middle ground between intact top-down proteomics and break it up to peptides, shotgun proteomics. And you'll sometimes hear this called middle-out proteomics. I wanted to note that antibodies, monoclonal, all antibodies essentially, have special kinds of clipping that are available to them that do not apply in other types of proteins. In this case, I'm showing you the enzyme IDES that is being used to separate the part of the antibody that's specific to the antigen sometimes called the Fab, which is at the top of the sequence here, the V at the top. It's separating that Fab from the constant region that can be clipped away from it. So if you're trying to discern just the specific parts of the antibody, you can do this kind of separation and make the problem a little bit simpler, because frankly, getting the entire sequence conformation from an intact monoclonal antibody is very, very difficult. Okay, so why is it that for so long people have focused on the study of peptides as the mainstay of proteomics? One of the key reasons for this is that dealing with the analytical chemistry in intact proteins is very, very difficult. So now we're shifting into the part of the talk where we assess why is it difficult to make top-down data. So I thought this quote from a 2018 paper by B. Chen et al. Was really valuable for this. Numerically, the intact proteome appears to be a much simpler mixture than its corresponding peptide digests. I mean, to prove that to yourself, you could simply say, if I'm using trypsin on this protein mixture, I'm turning on average 1 protein into 10 peptides. Therefore, the diversity of the mixture gets worse. In practice, however, protein level fractionation and separation are daunting tasks due to the diverse physiochemical properties, such as big changes in how large a protein is. I mean, insulin is going to be quite different than titin, right? Charge, insulin is something like a +7, but some of the larger proteins that we work with today in this field are charged more than 50 plus. So you're dealing with ions that are charged at a much higher level than are the peptides we typically deal with in shotgun proteomics. And hydrophobicity. Now this is a theme we're returning to, that an intact protein has on average a much greater hydrophobicity than do peptides. So if you stick them, if you're using hydrophobicity as an axis for separating these ions, you need to be sure that you can actually get the protein off of the hydrophobic materials at the end of the day. And of course, don't forget the wide dynamic range of the proteome. This has been a bedeviling feature for people in the shotgun field for a long time, because the study of biofluids in particular becomes quite challenging when the top 10 proteins are present in 10 times as many copies as the next 10, which are present at 10 times as many copies as the next 10 proteins. So the huge dynamic range that we experience is almost always going to be a feature that vexes us in the space of top-down proteomics. So let's talk about this hydrophobicity aspect for a moment. I want you to imagine that A is a big bead by comparison to the chains here. So what we're looking at are the materials that we put into a column when we are separating peptides, the C18 beads, or when we're separating proteoforms, the C4 beads. You can see then that the C number is just telling you how long is the chain, essentially, of these aliphatic sequences stretching away from the core bead. So if you're going to use this kind of separation on the basis of hydrophobicity, you need to use a material that is appropriate to the ions that you're actually separating. And in this case, it's certainly the case that proteoforms are going to require a much less hydrophobic material. That can also diminish the degree to which you get a good separation by this technique. I would just note that the equation at the bottom is principally important because we need to think about the fact that we're using a very high amount of pressure to cause these the solvent to flow through the RPLC column, the reverse phase liquid chromatography column. And how big the beads are is going to have a lot to say with how much back pressure we get. So it can be very difficult to push these solutions through to get a decent flow of ions. So this equation is just one way of capturing how this becomes complicated as we change the particle size and so on. This is one of the reasons why we sometimes see that people in top-down proteomics do away with their liquid chromatography. I've been working with a data set recently that uses capillary electrophoresis instead. So instead of using reverse phase liquid chromatography that separates on the basis of hydrophobicity, we're using capillary zone electrophoresis. This leads to a really big change in what the source looks like for these. And frequently, we have relied upon the use of reverse phase liquid chromatography to desalt our samples as well, to make them a little more amenable to going into the mass spec. So people have tried alternatives to hydrophobicity en route to the mass spec, and in some cases, the data come out pretty well as a result. There's a lot of innovation still taking place. I don't think that the book has yet been written that this is how thou shalt do top-down proteomics. We instead see a lot of diversity in what ideas people are bringing to bear to make this easier to do. I would also note that a lot of the assumptions that we make about mass accuracy for bottom-up proteomics do not translate particularly well to top-down proteomics. So let's imagine that we have a peptide of, say, 27 or 28 amino acids. You might imagine that it has a mass of 3,000 Daltons. And in bottom-up proteomics, something like that is likely to have a charge of about +3, which means that you see an ion at 1,000 m/z, 3,000 Daltons divided by 3 charge. So now we have our ion at 1,000 m/z. We typically, in a Fourier transform instrument, assume that we have measured its mass accurately to a level of 10 parts per million. So you can divide 1,000 m/z by 1,000,000, multiply that by 10, and now you get what the mass error, or what the mass to charge error is on that. Multiply that by the charge, and you learn how much Dalton error, how much mass error you have on the intact peptide. So one of these, the 10 parts per million refers to a mass to charge error. We multiply that by the charge to get to the mass error for the intact peptide in this case, or the protein in the lower case. So if we look at the bottom, we look at what is today a pretty challenging case for tandem mass spectrometry of proteoforms. Mouse tubulin beta is just under 50,000 Daltons in size, 50,000 versus 3,000. So much, much larger, many more peptide bonds. And because it's got so many more side chains on it, it's able to take on a charge of +50, 50. So even though both the short peptide we described and this long proteoform appear at 1,000 m/z, when you multiply the mass error by 50, you find that having a mass error of plus or minus 0.03 Daltons for the peptide at the same parts per million mass accuracy reflects a plus or minus half a Dalton error for the intact protein. That is a considerable problem. And I'm actually ignoring one of the nastier problems that comes along with this, that for an ion of this size, the number of isotopes you can observe for it is large. It's very large. How are you to discern just where the monoisotope appears when the average case is that you've picked up a dozen extra neutrons in the course of the sequence? It becomes much more difficult to say this is the monoisotope or that is the monoisotope. Very challenging. It may be that there's no observable monoisotope and you're just kind of guessing where it should appear. As a result, the mass accuracy information we have about these intact proteoforms is of considerably lower quality, and it's of much lower use in trying to discern which proteoform this tandem mass spectrum represents. And of course, we have problems of dissociation, that we want to break the peptide bonds for ions of a particular proteoform in order to produce a tandem mass spectrum. That tandem mass spectrum should inform us about the sequence of whatever broke to produce it. But to do that means that we need to break quite a few of the peptide bonds to be able to produce terminal ions, B and Y ions if we're doing something like CID, or C and Z ions if we're doing something like ETD. To do these, to find that we have enough of these breakages that we can reliably assign a sequence to a tandem mass spectrum is a big deal. And frequently we find that instruments are set up in kind of a switch hitter mode where every ion gets analyzed both in collisional induced dissociation and in electron transfer dissociation in the hope that one or the other of those will be identified. It's quite a difficult challenge because when you have hundreds of peptide bonds versus a couple dozen, your ability to break a variety of them actually diminishes, and you frequently find that you're relying on fewer ions to make your assertion about what this sequence or what this tandem mass spectrum actually matches. So you will frequently find that in the space of shotgun proteomics and bottom-up proteomics, you quite frequently observe that people are labeling tandem mass spectra with B and Y ion pairs or C and Z ion pairs if we're using ETD. So something over at the right in this slide is quite a common representation. But I want you to note that this annotated MSMS information that I'm showing is actually abstracted from the larger spectrum below. They're both the tandem mass spectrum, but one is only showing you about 10 m/z drawn out of this much larger spectrum. And look how crowded this is. We have things like the Z57 ion, which is a C-terminal fragment of a protein that contains 57 amino acids, here charged +3. We have, say, the C80 ion. This is an N-terminal fragment that contains 80 amino acids. It's charged at +4. And we also have things, if you look at the left of that little spectrum, we have a C120 ion. So it's 120 amino acids from the N-terminus, and it's charged at 6+. So you can see then that we have all of these different charges for all of these different fragments appearing, and each of these is decorated with a whole bunch of different isotopes. The number of fragments that result from the breakage of a top-down proteoform is enormous, and it's quite difficult to assign them all with a high degree of accuracy. As a result, people do not frequently visualize their tandem mass spectra in this annotated way. Instead, you'll frequently see what's called a fragment map in the middle of the slide here. So when you see a little hook sitting between a pair of letters there, that is to say that we have direct evidence of a breakage this far from the N-terminus that would lie between these two amino acids that confirms our assessment of what the sequence should be. Now, I want to note that this is a gorgeous identification that's being shown in this demonstration fragment map. More typically, you're dealing with a dozen or 2 dozen peptide bonds that have evidence in the tandem mass spectrum. So it's not often the case that you get a really beautiful one that's going to tell your biological story. You're typically relying on far fewer matches to the peptide, the polypeptide sequence. And of course, frequently people are trying to answer quantitative questions about their data. And for this, they may end up highlighting data from the MS scan, not the MSMS scan. So the MSMS is the set of fragments seen for the proteoform. The MS scan here is showing us the unfragmented proteoform ions themselves. Okay, so those frequently have these isotopic envelopes that show all the different isotopes that we're seeing for each of these intact proteoforms. So if these are the kind of data that we're producing, how do we get from one to the other? I would start with the fact that deconvolution is hugely important in this space. You know, in shotgun proteomics and bottom up, we often think about the database search engine as the thing that needs all of the attention. But in the top-down world, deconvolution is a very big deal. And there are 4 rather different goals that these tools all try to solve, and I think it's important to walk through them one by one. First off, how can we combine together the charge variants that are seen for each fragment to a neutral mass? It may be that the C50 ion, one that contains 50 amino acids from the N-terminus, shows up in charges +20, +21, +22, +23, +24, +25, several different charges. So this is the same chemical composition, but it's showing up at different charge states. And those charge states can be combined to say, oh, let's drop all of these to a neutral mass and put one isotopic packet in to represent all of these different charges of the fragment that are seen. So that's one of the first stages that we're going to need to do with each tandem mass spectrum. We will also want to shift intensity from the more massive isotopes back into the monoisotope where that's possible. So if it's possible to say these 4 variants of a fragment ion all represent a ladder, each larger than the last by 1 neutron because it has 1 more C13 in it. So now we can say, well, let us deconvolve this information back into a single peak. So in the first stage, we were combining charge variants of the fragment. In the second phase, we are instead trying to put all of the isotopic variation back into a single peak. Certainly, being able to make assessments of the original precursor ion on the basis of mass and charge is something that we can refine here. Just as an example, when I run an instrument on a thermomass spectrometer, I might expect that 25 plus is the highest charge that the instrument will ever automatically assign to a precursor. And yet the true charge might be higher than that. So I can refine this precursor mass and charge after the fact, and in many cases, rescue a tandem mass spectrum by fixing its precursor. And of course, being able to integrate the precursor intensity is a rather big deal if you're trying to do anything about quantitation, to say that this proteoform is a more frequent variant of this protein than another isoform. Okay, so much of that falls under the heading deconvolution. Right now, there are 4 different search engines that we see used very, very broadly. I think it's probably worth a moment to just sort of talk about what these look like. ProCite Proteome Discoverer is an algorithm that was initially published by Neil Kelleher's group many years ago. More recently, they've created a company called Proteomics that handles the marketing and the support for this tool, which means that if you purchase a license to use ProCite Proteome Discoverer from Thermo, the company that makes it available, you are entitled to support. You as a graduate student can write them saying, my lab license number is such and such. When I analyze my data set, I get this error, please help. And you have some chance of getting a response from them. So that's a pretty nice thing. A lot of service facilities who want to use top-down proteomics rather than conduct research in top-down proteomics need that kind of service and support. More frequently, we have seen that people have opted in recent years to make use of some of the free alternatives to ProCite Proteome Discoverer. Those would be Topic Suite, MS Pathfinder T, and PTOP. All of these are free to use. Now, I wanted to note that at present, the source code for PTOP does not appear to be available. So if you are a computer programmer and want to mess around with the code, maybe fix bugs that you find, that's going to be an option for you with MS Pathfinder T and with Topic, but it's not yet possible within the context of PTOP. So Topic Suite is one that has a whole lot of really unique features, including the routine ability to spot mass shifts that correspond to post-translational modifications, even if you don't know in advance which ones you're looking for. So that's a very special ability within Topic, and it offers some other nice features like the ability to determine how much information you have about the true location of a given post-translational modification. So I tend to think of it as kind of the Cadillac of this set. On the other hand, I frequently find that I get more identifications in the form of proteoform to spectrum matches when I use MS Pathfinder T or PTOP. On the other hand, these two fall on a very wide spectrum. PTOP is very, very quick. MS Pathfinder T, on the other hand, can take as much as 4 times or 5 times as long to run on exactly the same data as does PTOP. PTOP and ProCite Proteome Discoverer are the fast ones here. Topic and MS Pathfinder T are the slow ones. And we would also ask, how well do these things work? I'm actually in the middle of writing a manuscript about this. So this is a figure that's come out of our work in these early stages. What we did was to run the same data sets through 4 different search engines, looking for exactly the same modifications, using exactly the same sequence database in each case. And then we asked, of the peptide spectrum matches that the proteoform spectrum matches that resulted, what fraction of them were identified by all 4 of these search engines, by 3 of the 4, by 2 of the 4, or by 1 of the 4? And when we look at the results from zebrafish, D. rario, the top line there, or E. coli, we see that approximately 30% of all identified spectra were identified by all 4. That is a pretty good result. On the other hand, when we looked at the data from Cephalobus islandicus, we were looking at HCD data sets and ETD data sets separately. We saw that the fraction of spectra identified that were identified by all four was a lot lower. And frankly, to get the number for zebrafish up as high as we did, we had to switch from using ptop1 to ptop2 because there was a problem with the way that ptop1 handled that data set in the earlier case. So if you work at it, you can get some degree of agreement among these search engines, but it's not going to be the case that every identified spectrum is going to be identified by any of these search engines. Frankly, using multiple search engines is quite necessary to getting to a place where you can identify a solid fraction of these data sets. There is no perfect search engine. All of these are going to have trade-offs. So if you're going for a fast answer, ProCite, Proteome Discoverer, and ptop are your friends. They really are much, much quicker at coming up with answers than are the other two search engines. If you're looking for an answer where every proteoform listed is one you can rely on, I have to say ProCite Proteome Discoverer does a better job of filtering out false positives than anything else. In fact, if you tell it to give you a 1% false discovery rate, you're likely to get back something closer to a 0% false discovery rate. So it's a very specific engine. The flip side of that is that if you're trying to explain as large a fraction of your spectra as possible, you really need to think about which ones are sensitive search engines, not specific. So for this, MS Pathfinder T and ptop2 have been very, very good. If you include unknown modification matching, Topic is also very good at high sensitivity. I've been throwing those out in my analysis, and as a result, it doesn't look as sensitive anymore once you eliminate its ability to use unknown mass shifts. If you're unwilling to pay any money, ptop, MS Pathfinder T, and Topic remain good options for you. They're all free. Are you always going to get quick feedback when you ask people for help with software? Not necessarily. MS Pathfinder T and ptop have not really got active development going on with them right now. As a result, your ability to get feedback on what's going well or poorly here may not come with a quick answer. If you're trying to make a very thorough appraisal of your data and allow for the possibility of unknown modifications, Topic and MS Pathfinder T are quite good. I think that people often overestimate their willingness to dig through the nuts and bolts of the feedback that software gives them. And if you're not willing to do that, Topic is not really your friend. Interpretability is probably going to be a bigger aspect for you, and the simpler output that you get from ProCite Proteome Discoverer, from ptop, and from MS Pathfinder T is probably going to be a little easier for you to deal with. So let's talk about lowering the bar. I'm really excited about the future of biomedical mass spectrometry across Africa. And back in 2018, I teamed up with a bunch of labs across South Africa to ask about how widespread are biological mass spectrometry groups in the country. And in 2018, there were already 18 laboratories that would allow shared use of their instruments. 18 labs in just one country. Now, granted, South Africa has a much better biomedical research structure in place for biotechnology than a lot of places. But I am glad to see that there are public services in nations like Nigeria and Kenya, and I believe also true for Egypt and some other countries, that will allow us to send off samples for mass spec analysis, even if you're not doing it in an institution in your own country. So it is possible to do a send away scenario where you send your sample to another country in Africa or to just another university within your own country to get biomedical data back in the form of mass spectrometry data. So that's comforting. This technology is coming, and it isn't coming everywhere immediately, but it is happening slowly, that biomedical mass spectrometry facilities are opening their doors across Africa. That's very comforting for me, because this is the field where I want to be employed in Africa again. I'm up here in France for a couple of years because of the COVID pandemic, but I really do hope to come back to be a professor in Africa, or South Africa, hopefully, again, very soon. But this, at the moment, the laboratories I'm familiar with within South Africa are all doing shotgun or bottom-up proteomics or they're doing metabolomics. At present, I have not seen laboratories doing top-down proteomics, where they are fragmenting intact proteoforms for analysis. This is something that we can correct. This is something that can be fixed. And happily, there are a lot of bottom-up laboratories that have a good investment in the technologies of shotgun proteomics that are able to make this jump, I believe, to do top-down proteomics as well. So what's stopping us from that? Certainly, right now, the technologies for separation and ionization have been very much specialist emphases. There are some broad rules that we can work with. We know that C4 separation en route to electrospray ionization works pretty well. But if you're going to be able to deal with just any sample someone brings you, you're probably going to need to have a specialist who's able to evaluate different techniques that may be used for this separation upstream of the mass spectrometer. I wanted to note that a lot of the data sets I'm working with from North America and from Europe at this point are using very high-end thermo instruments, instruments like the Orbitrap Eclipse. And if you were to buy just an Orbitrap Eclipse today with no pumps or plumbing, just the mass spectrometer itself, you're likely to spend $1 million. That's rough. It's a tough ask for a lot of institutions in South Africa or in other countries in Africa to spend $1 million in that kind of outlay. In fact, when I look at the website for my own university's proteomics laboratory, I see that they're still talking years later about how they had the first Orbitrap Velos in South Africa. They had the first fusion, Orbitrap fusion in South Africa. So there's a lot of effort, I think, in buying prestige instruments, but I think that we can pay attention to the fact that you don't have to buy the most expensive possible instrument in order to do quality top-down proteomics. So I would point to instruments like the Q-Exactive HF. The Q-Exactive is already available at several campuses across South Africa. The high field variant, the HF variant, is particularly useful when you are studying very large ions and need to resolve things very, very well. So a Q-Exactive HF is not one of these $1 million instruments. It's a less expensive one that can still do very high quality tandem mass spectrometry of intact proteoforms. I am working with data from the QE-HF daily, and I would say it's a very impressive instrument that does not necessarily empty the bank account. That said, you do not have to buy a thermo instrument in order to do tandem mass spectrometry of intact proteoforms. I've been doing work now with a variety of data sets from Yin Gu's team at the University of Wisconsin. Her lab uses a lot of Bruker QTOF instruments, which is to say a quadrupole does a selection of the precursor ions, and then a time of flight cell does the tandem mass spectrometry on them. So this Bruker QTOF instrument that they frequently use is a Maxis 2. It's not even the most recent model of what Bruker makes in QTOFs. That would be the timsTOF. But it is possible to buy these less expensive QTOF instruments, much as you would buy a Q-Exactive HF, and do very high quality proteoform identification. As I mentioned, the software for doing deconvolution and identification can be simply free. You can just use free alternatives. Frequently, you'll see as good a result as you will from the pay-for software. On the other hand, the support you receive may not be as comprehensive. Interpreting top-down, I misspelled interpreting, I feel so bad, sorry. Interpreting top-down identification and post-translational localization, trying to discern which amino acid is carrying a post-translational modification, is difficult. It is difficult to curate these very closely. However, if you've already developed skills in bottom-up mass spectrometry and peptide-based mass spectrometry, you've already developed some skills that are relevant to doing this at the top-down level as well. And at base, you can think of the biological equipment that will allow you to break into this field at a more basic level, frankly. At present, there's a lot of emphasis, at least within South Africa, on having the best instrument and the best scientists and so on all pulled together. And we lose track of the fact that a lot of people want to be proteomics users, not proteomics technology developers, which means that we often overlook how much value we could get from a less expensive, often easier to maintain instrument. So I'd already mentioned Q-exactives and Q-TOFs. They are certainly great instruments. At that picture below, you can see an example of what a Q-exactive looks like. And the two black boxes that are closest to the camera are pumps. We use those pumps to drive the liquid chromatography. So it's not a huge instrument. It's not going to require a room of its own, but it is still a substantial investment in money. I think that a lot of people miss out on the opportunity of quadrupole ion traps. Instruments like the LTQ, for example, from Thermo, or the Bruker Amazon, or traps available from other companies, can give you really good proteome identifications for the bottom-up experiments. And I have to say, at the moment, South Africa is having some difficulty keeping the lights on continuously. So what happens to an instrument that needs to have a very high vacuum inside if suddenly the lights turn off, right? That's not great. It's true that a lot of medical centers have generators that are at least supposed to kick in to bring to keep the power on steadily, even when the power turns off. But if you're trying to maintain a very high-end instrument under these circumstances, things are going to be very difficult. And I would point out that things like a quadrupole ion trap that I show in the middle, or a triple quadrupole that I show over at the right, is actually much more robust to these circumstances where the power is flickering on and off. So I propose that a lot of laboratories could actually benefit by having an ion trap and a triple quadrupole for handling identification and quantitation experiments, then would benefit from spending a lot more money on something like a Q-exactive or an Orbitrap class instrument or a Q-TOF. It is possible to spend less money initially on the instrumentation and still get quite a lot of bang for your buck. So top-down proteomics evaluates these samples at the level of proteoform rather than the peptides that get digested from them. You don't have to do protein assembly to infer a protein list from the peptides you've identified if you're doing top-down proteomics. Different preparation methods will certainly apply, as will the different separation methods you need, and perhaps even the different fragmentation techniques that you need in order to shift you to top-down proteomics from bottom-up proteomics. But at least I feel comfortable saying enough software infrastructure is now in place to answer biological questions, not just technical questions about how do we make this technology work. Instead, people are now winning awards for the biology they're learning because they have top-down proteomics technologies available. I'm very grateful to Epic XS for funding me to do this work up here in Paris. But I gotta say, I'm really looking forward to coming home to Africa. I will have been away for 2 years at the end of 2022, and I want to come back. I love what I've found. I love the life I've found in Africa. But that said, I definitely want to thank Julia Shomoruk, who took a chance on letting me come up to be a visitor in her lab for a year, and now for 2 years. I think that we're doing great work together, and I'm very excited about the things I've gotten to learn as part of her group. So, and of course, thank you to H3 Africa for inviting me for this talk, and I'd be glad to take any questions we have time for.
Okay, this was really amazing, David. Thank you so much for the presentation. So yes, now time for question, comments, and answer time as well. So please do raise your hand if you have something to say, or tap in the chat box as well. David will gladly sort of engage with you. We have about 2 minutes, but I can extend it by 5 minutes just to make we give everybody enough sort of time to sort of engage with David. But just to say, this is live on Facebook. I'm recording it, and I will share the recording in the slides with you. So I'm going to put it up on YouTube, and I'll share the links with you through our mailing list and social media as well. But yeah, it's time for questions and comments. I'll be looking at the chat box. And you can raise your hand, and I'll allow you to sort of unmute yourself. Okay.
I just got to say, I always love this part of the talk, so I'm happy to answer any question, big or small. And questions about science careers are also fine.
Okay. Thank you so much for that intervention, David. So, I mean, from my side, you mentioned South Africa a lot because I think maybe that's because you were based in Stellenbosch. But I just want to understand what is the situation in Africa, other African countries, not just South Africa, in terms of obviously resource challenges. Because I mean, I know one of the things that you keep on mentioning is the funding or resource issue. I mean, there's 18 labs in South Africa. What's the situation across the whole continent, but not just Africa, but other LMICs as well? And how do we sort of navigate and mitigate the resource limitations? Because I mean, obviously, as you said, I mean, one of the machines cost like $1 million US, and that is a lot of money that a lot of sort of LMICs would sort of struggle to even raise those funds. So how do we sort of mitigate that as well?
Well, as I was saying, there's a lot of biology that one can accomplish even with a less expensive instrument. When asking how widespread is biomedical mass spectrometry in Africa, getting current information that's currently relevant can be really difficult. Even for my own university's proteomics laboratory, they are advertising that they have an Orbitrap Velos, but the Orbitrap Velos hasn't been working for 2 years. As I did my survey across South Africa, I found frequent cases where the information that I was seeing on the web was very much out of date. So I haven't yet done this kind of survey for example, Egypt or for Kenya or whatever. But I think that many laboratories in Africa are currently making use of technologies like GCMS, which is frequently a much less expensive model than even the ion traps I was talking about. This is gas chromatography, electron impact ionization mass spectrometry. It's used for recognizing volatile compounds. So people who are doing some types of metabolomics research could use GCMS. But we don't really think of that typically as biological mass spectrometry. Typically, that means being able to deal with things like sugars or lipids or peptides, things like that. Generally, those don't break apart very well in electron impact, nor are they very separable by GC. So to say what constitutes a biological mass spectrometry lab answers lots of different questions. And finding this information without being in the country can mean that I can be led astray by a website that's been out of date for 3 years. So it's difficult to collect that information broadly.
Okay, thank you so much for that answer, David. There's a comment from Samuel Eziozo saying, thanks, David, for your talk on proteoforms. It will be an interesting imaging analytical tool for intact proteins analyses. And that's from Samuel.
Thank you. Yeah, I agree. This is important. As somebody who's been in mass spectrometry for 20 years, I've seen a lot of technologies that make a huge splash, and then I see that over years they remain a boutique technology that not a lot of people actually implement and use. And I think that Topdown has turned the corner on that. It's not a boutique technology. It's now something that a lot of universities want to be able to use regularly. So I'm excited to see that about top-down proteomics.
Okay, thank you so much for that. I have time for one more question or comment because I also have another meeting to go to now.
Sorry.
Thank you, David, for this good presentation on proteoforms. So I'm glad people enjoyed this. And there's more people trying to join the room now, but we are just about to finish, okay? But David, thank you so much for your time. I'm really grateful that when I reached out to you to ask you to give this webinar, you were really excited and you gave such an informative webinar. And I'm going to encourage people to follow you on Twitter. I'm going to encourage people to follow your blog and to find your YouTube videos, which obviously are amazing. And I mean, we'll teach them more about proteoforms. So you are actually the uncle, the go-to uncle for this. So much for your time. So yeah, for everybody, thank you so much for joining us for the first webinar of 2022. And we will have another webinar in February sometime. I'll send out ads for that. But for today, I just want to appreciate your support and that I'm hoping that we're going to have a long and safe 2022 so that we can obviously have monthly webinars as well. But to David, thank you so much for your time. Enjoy the rest of your day, everybody. And I'll speak to you next month when I advertise the next webinar as well. Goodbye, everybody. Thank you so much. Yes, you will have the PowerPoint and the slides. I will email that today or tomorrow. I still need to download it and upload it into YouTube and all the other stuff. But after my other meetings, I will send it to you guys, Odu. Thank you so much. Goodbye.